Hello everyone, welcome to today's live broadcast, Sequence Function Mapping to Determine the Impact of Coding Variants. I'm Rachel Brown of LabRoots, and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Doug Fowler. Dr. Fowler focuses on developing and implementing new techniques to address difficult problems in protein science and genomics. He is currently an assistant professor of genome sciences and affiliate assistant professor of bioengineering at the University of Washington in Seattle. Doug did his graduate work at the Scripps Research Institute, where he discovered and characterized the first mammalian functional amyloid protein. During his postdoctoral work at the University of Washington, he began to develop high-throughput methods for analyzing the effect of mutations on protein function. I will now turn it over to Dr. Fowler for his presentation.
Hi, thanks so much for that kind introduction. I'm excited to be speaking with all of you today. This is a new format for me, so thanks for, if I make any mistakes, thanks for bearing with me. Today I'm going to speak about a problem that my lab spends a lot of time thinking about, which is how to connect the sequence of a protein to its function and what implications that connection has for variants that we find in human genomes. So the first point that I want to make is that coding variation is pervasive in the human genome. This is a plot from a 2012 paper where 16,000 exons were sequenced, or 16,000 different genes were sequenced in about 2,500 individuals. And what you're seeing is the number of variants per individual on average in coding regions. And what you can see is that first of all, there are lots and lots of coding variants. So that's the first thing. The second is that if we break those variants out by type, we see that the sort of easy to interpret ones like splice variants and nonsense variants, where we can reasonably assume there is likely an effect on the protein, and the synonymous variants, where we can reasonably assume that there's likely not an effect on the protein, comprise only about half of all the variants that we see. And in fact, missense variants comprise a pretty large fraction. And missense variants are interesting because a missense variant could range from having the effect of complete loss of function to being in fact neutral. One additional interesting thing that came out of this work, and has since been sort of validated by many others, is that much of the variation that we see is rare, and in particular the variation that might be functional or might be damaging. That's overwhelmingly rare. So what's true is that interpreting coding variation is both challenging and important. And it's important because now it's really easy and quite cheap to sequence a human genome, and people are, as sequencing moves into the clinic and direct consumer sequencing continues to grow, people are left with this question. We sequence a genome, what we find there that's interesting is variation, right? And people want to know, what do these variants do? What do they mean? What do they mean for whatever disease or phenotype that I'm interested in? And so that's why interpreting variation is important. Now, making the connection between a variant and a phenotype is also hard, and it's hard for a number of reasons. The first is that what we'd like to do is sort of gold standard genetic analysis on all the variants that we find in genomes, but that requires that the variant be fairly common, such that we can collect a reasonable number of people with the phenotype and without, and with the variant and without, and then associate the variant with the phenotype. But that's really hard to do when variation, when we're talking about rare variants, because we just can't collect enough people to do that type of association. So these sort of rare variants, which are numerous and really hard to interpret, are a problem we've thought a lot about. And there is a way or there are a number of methods that can be used to interpret these variants. The one of which we're sort of interested in, which we're going to focus on today, is by making those variants and then seeing what they do to the protein. So this is, people have done this for many decades. You can express and recombinantly purify a protein containing the variant in question, or you can edit a cell line such that the cell line has the variant that you're interested in, and then see what happens. And so these mutagenesis experiments are a great way to interrogate functional effect, but they do have one big limitation, which is that, at least as they're traditionally practiced, they really don't have a hope of addressing the vastness of sequence space, right? So if we think about a prototypical human gene, which might have 7,000 variants, possible single mutations, so possible singly mutated variants, a person could walk into a clinic or get their genome sequenced and find any one of those 7,000. But there are a finite number of functional experiments that you can do. You can only synthesize so many proteins and study them. You can only make so many cell lines and study them. And so while these approaches are great, they are sort of limited in their ability to be deployed. And then there's another problem, which is that for all those, there are 20,000 genes in the human genome. So we've got 7,000 variants by 20,000 genes. It's a really large number. So that's sort of the problem that motivates us. And I'm going to tell you today about three distinct approaches to sort of getting at this problem. So the first is deep mutational scanning, which is a method that we developed and are continuing to work on, which allows us to interrogate the functional consequences of variants on a very large scale. The second is Envision, which is a predictor of variant effect that we've developed that can be applied genome-wide, and which leverages this large-scale mutagenesis data that we and others have been collecting. And then finally, I'm going to tell you about a tool that we're developing that can allow us to make libraries of variants at multiple loci in the genome, which addresses a sort of an additional important sort of biology and genomics question, which is how variants interact when they're at multiple loci. So that's a sort of outline for the talk today. So let's dive right into deep mutational scanning. So I'm going to talk you through a prototypical deep mutational scan here. And the idea of a deep mutational scan is to experimentally measure the functional consequences of hundreds of thousands of variants of a protein by marrying selection for the protein's function to high-throughput DNA sequencing. And a deep mutational scan starts with a DNA-encoded library of protein variants. I'm showing you an example library here with 4 variants, each one a different color. And we need to instantiate this library in a system where the DNA encoding the variant is linked to the protein variant itself. So a simple example of this would be a culture of cells where each cell gets a different plasmid, and each plasmid carries a different variant. But there are other examples that we'll talk about that satisfy this requirement. So once we've got our variant library, we then impose a selection for the function of the protein that we're interested in. And this selection could be anything from asking the protein to bind a partner to complementing a deletion of this protein in the genome. And imposing this selection perturbs the frequency of variants in the library according to their ability to perform the function. So here you can see that the red variant has increased in frequency, presumably because it performs the function that we selected for quite well, whereas the blue variant has decreased in frequency because maybe it's a loss of function variant. So then the next thing you do is come in with high-throughput DNA sequencing and measure the frequency of each variant in the selection, both at the beginning and at time points throughout the selection, and then at the end. And so what we've shown is that you can take this high-throughput sequencing data and develop a functional score from it that recapitulates the biochemical effect of a mutation on the protein. And so This is just one simple way of calculating a functional score, and that's to suppose, or to calculate an enrichment ratio, where the ratio is the frequency of a particular variant after selection divided by the frequency of that variant before selection. So if the ratio is greater than 1, as is the case for the red variant, you would infer, or we would infer, that that variant contains mutations that enhance the function of the protein. Whereas if the ratio is less than 1, as is the case for the blue variant, then we would infer that that variant contains mutations that are deleterious to the function of the protein. Now, we and others have developed more nuanced ways to generate functional scores, including taking multiple time points and regressing, or using flow sorting to sort variants into bins, activity-based bins. We have a software tool called Enrich that we're about to release a big update to that can do a lot of this analysis. So anyway, that's deep mutational scanning. The point that I want to make sure everyone appreciates is that this process can allow us to ascertain the consequences of hundreds of thousands of variants for the protein that we're studying, which means that we can cover all of single mutant space and get pretty far into double mutant space, which is a useful thing to do. So I'm going to tell you about a couple of quick examples of deep mutational scans, just to sort of cement our understanding of the method, because a couple of the future points of the talk are going to depend on it. So the first deep mutational scan we did was of a small domain called a WW domain, and you can see one here. It's this three-stranded beta structure in blue, and WW domains in nature mediate protein-protein interactions and in vitro bind peptides, and that's what you see in the structure. So we mutagenized a 34 amino acid region of this WW domain, introducing 2 mutations per clone, and then we cloned the library as an in-frame fusion to a phage coat protein and did a phage display experiment or selection where we asked that the library bind to the target peptide for this particular domain over several rounds of enrichment. We then sequenced all of those rounds and calculated a functional score for about 100,000 variants that were present in our library. We can look in the data and pull out all the mutations, single mutations to alanine. So if we plot those the effects of those single mutations here, on the x-axis you have position along the mutagenized region. On the y-axis you have the log transformed functional score. So the gray bar is equal to what the wild type score is, and each blue point represents a measured functional score for a particular alanine substitution. So this sort of recapitulates an alanine scan. Of course, we have the functional effects of most mutations at most positions, so we can construct a sequence function map, which is just a visualization or visual representation of the effects of all the single mutants on this domain. And in the upper left-hand corner, you have the alanine scan that I was telling you about. Next door is cysteine, aspartic acid, and so on and so forth. So this is sort of a visual way to see exactly what the effects of all the mutations you could make, or most of them, in this domain are. And there's an interesting biochemistry story here, and so on. But in any case, we and others have shown that this type of data, if you set up the experiment right, correlates very nicely with measures of the protein activity that are developed using traditional, biochemical methods. So another deep mutational scan that I'll tell you about that sort of presages or tells you a little bit about the direction that we're hoping to head, or that we are heading, is one that was conducted principally by Leah Starita. This was a collaboration with Jay Shanduri and Stan Fields. So Leah is interested in BRCA1, which is a tumor suppressor that's important in breast cancer. It's been studied for many years, but there are still a number of women whose BRCA1 gene is sequenced and who are given a diagnosis of a variant of uncertain significance. That is a mutation that's never been seen before and about which we don't know really much of anything. And so our idea was to conduct a deep mutational scan of BRCA1 and then to see if we could say something about the variants that are observed. And another interesting thing about BRCA1 is that it's a multifunctional protein. So it sort of mediates DNA damage repair. That's its sort of high-level function. But it's also an E3 ubiquitin ligase and binds to a number of binding partners. And so our idea was to decompose the multiple sort of functions of BRCA1, or measure the individual functions of BRCA1, and then combine those deep mutational scans into a model that would be predictive of this DNA damage repair function, which is important for tumor suppression. So Leah developed a phage-based E3 ubiquitin ligase assay, and then used deep mutational scanning to construct a sequence function map, where the function in this case was the E3 ligase activity. She also developed a yeast 2 hybrid-based BARD1 binding assay, where the BRCA1 was being asked to bind to BARD1, which is a critical binding partner for BRCA1 in vivo. So we were armed with these two deep mutational scans, and then we'll get to the modeling in a minute, but first I want to show you what the sequence function maps look like for these scans. So here you have on the top the E3 ubiquitin ligase sequence function map, and on the bottom the BARD1 binding sequence function map. And these maps are organized in a slightly different way than the one I showed you before. They're heat maps, where each column is the position and each row represents a possible amino acid mutation that could be made. And the more red a box is, the more highly functional that mutation is, and the more blue, the less functional. So what you can see here is that the two scans, or the two functions, have some similarities and also some differences. And in fact, there's a whole interesting sort of biochemistry story here that part of which has been published and part of which we're working on, that tells us about how BRCA1 sort of works. But to get to the point that I was talking about before, which is making a model to predict DNA damage repair in BRCA1, what we did next was gather data on DNA damage repair in a human cell model for 45 mutations, and then train a predictive model that combined both the E3 ligase and BARD1 binding data to predict the DNA damage repair data. And what you see here is a plot on the x-axis of the predicted DNA damage repair in the assay, and the y-axis, the observed score. And the red points are the known pathogenic mutations, and the green points are the known benign mutations. And what I hope you can appreciate is that our model separates the benign from the pathogenic quite well. In fact, it performs better than any other predictive algorithm, computationally rooted predictive algorithm out there. So this sort of gives you a taste of where we're hoping to head with deep mutational scanning. I will make the point that it's a maturing technology. So I told you about a couple of examples, but in fact there are many. These are just some of them now. You can use model systems ranging from phage, which we talked about, through coli and yeast to mammalian cells. The size of proteins that can be mutagenized has grown and is now up to larger than the size of a typical human protein, about 500 amino acids. So that's very satisfying. In my lab, we're working on a number of different sequence function mapping efforts. One, to look at resistance to targeted cancer therapeutics. Two, to examine other tumor suppressors. And here we envision doing deep mutational scans on the 5 or 10 most important tumor suppressors, and then using that data to try to develop genetically rooted prognoses, prognosis. And finally, we're looking at the activity of pharmacogenomically important enzymes, where we hope to be able to predict dosing requirements based on variation that we observe. So that's sort of what we're doing with deep mutational scanning, and hopefully it gives you a sense of what the technology can do. But it still leaves us with a big problem, right? Deep mutational scans are great. We can comprehensively profile the effect of mutations in a single protein, but I hope you can appreciate that developing an assay and making a library and doing all this sequencing, it's expensive and it takes time. And although we're dreaming of ways to do deep mutational scans on a sort of organism-wide level or a cell-wide level, or at least many genes simultaneously, we can't do that yet. And so we're still left with a problem when we think about interpreting variants on a genome-wide scale, which is that we can't get to all the genes experimentally. So that's what got us interested in developing a computational predictor of variant effect, and so that's what I'm going to tell you about. That's what I'm going to tell you about next. So variant effect predictors have been around for quite a while. This is just a partial list, or a graphic showing a partial list of variant effect predictors. In general, these predictors can do fairly well at predicting the effects of mutations, both in coding regions and non-coding regions. And they sort of work in the following way. So this is this is a panel from the Polyphen paper, and it sort of just lays out how the predictor works. They're often, very often, machine learning type approaches where a training data set of variance is gathered, and that training data set is labeled. Often, or pretty much all the time, it's labeled it's labeled in a way where there are deleterious variants and there are neutral variants. So then the model learns a way to classify variants as either new variants that aren't in the training data set as either neutral or deleterious. The way that's done is by attaching to each variant a set of features that inform that the model can then use to make predictions on variants it hasn't seen based on the feature values for that new variant. So that's how these predictors basically work. And what we observed was that they're generally trained on these very large, very sparse data sets, right? So this is the Polyphen example is instructive. There are about 50,000 mutations here in the Polyphen training data set, and they're scattered throughout the genome, right? So each gene, in general, would get 0, 1, or 2 training mutations in it. And if you think about it, that's a very different type of data than deep mutational scanning data. So the large-scale mutagenesis data you get from a deep mutational scan, it's for one protein, but it's very deep, sort of comprehensively tells you what mutations do to that protein. And so we sort of got excited thinking about this and thought that perhaps we could bring something to the variant effect prediction space by training a predictor on large-scale mutagenesis data that's quite different from training data that's been historically used to train these types of predictors. So our objectives in this project were to collect the published deep mutational scans that other people have done, add them to our own, and then annotate all the variants in these scans with descriptive features that we could use to train a model, and then in fact generate predictive models based on the data and see how they do. So I hope the sort of first part of my talk convinced you that we're sort of living in an age of high-throughput biochemistry where we can measure the effects of mutations on many, at a very large scale. And this first plot here is just the cumulative number of single mutations collected by deep mutational scanning approaches in the last 5 years, and that's rapidly climbed to 35,000. If you were to include multiple mutations, then this plot would go into the millions, but we restricted ourselves to single mutations here because we didn't want to be confounded by the effects of multiple mutations in training our predictor. So the proteins involved in composing this data set are shown here, where you see the protein by the number of variants on a log scale in each data set. For reasons that I'm not going to go into too carefully, the ones in blue are the ones that we chose to carry forward with as training data for our model. And I want to point out here that it's a bit of a rogues gallery of variants, of proteins. The proteins come from a variety of organisms, ranging from phage to humans. There are even some designed proteins that have been scanned, and they are also all done by, or not all, but mostly done by different labs. They're mostly done in different model systems. So some are scanned using phage display, some are scanned in yeast, some are scanned in mammalian cells. And many of these scans were done at sort of the very early days of deep mutational scanning, when sort of standards for generating and analyzing the data were less strong than they are now. And that means that it's like I said, it's a rogues gallery of data. So our thought is that if we can get something useful from this data as a training data set for a predictor, then things will only get better as we get more data at more deep mutational scans in the future. So anyway, that's the training data. So there are 11 proteins in the end that we felt that we could use, and this is just it looks like it didn't come through too well, but hopefully you can get the point. These are the 11 proteins that we chose, and these are just the distribution or histogram of the functional scores for each of the proteins. There are 20,000 total mutations in this data set, and the first thing we did was we centered all the data at 0 on the wild type, so everything is centered relative to wild type for each of the scans. And then, because we wanted to be able to compare between and sort of use as collective training data all of these different and sort of differently shaped distributions, we did quantile-quantile normalization to sort of equalize the shapes of the distributions and allow us to make comparisons between and across data sets. So That's the training data. We added descriptive features that are not particularly different from ones that other variant effect predictors use. There's a deep literature of sort of what works here, and we went with what works. We chose a variety of biophysical features that include facts about the wild type and mutant amino acid. We also, when available, use structural information. So I'll point out that we generated models both that use structural information and ones that don't, so we can make predictions on proteins without structures. But when it's available, we use things like the local density around the amino acid that's being mutated, the secondary structure, and so on. And then finally, we include biological features like evolutionary conservation, which we know to be a very important one in other predictors of variant effect. And then finally, we added domain annotations from UniProt. And the reason why we added domain annotations is that we imagine that in the future, as more and more deep mutational scans are constructed, we'll be able to make domain-specific predictions that are highly accurate, right? Because once we have a scan for a particular domain, any homologous domain in a different protein will likely have a pattern of mutagenesis or tolerance to mutations that's fairly similar. So that's why we included domain information. Okay, so as a first sort of test of the way we were training our model and the features that we chose, we decided to make individual protein regression models. And what I mean here is that rather than mashing all the data together to train a global model, we're going to try to train a model just on one deep mutational scan for one protein. And when I say regression, what I mean is we're trying to train a model that can predict the continuous real-valued functional score that we saw in the scan. So not a classification model like most other predictors that can separate damaging from neutral, but a model that can actually tell us what the score would be. For each mutation. So this is actually kind of a hard problem if you think about it. So we weren't sure what would happen here. And what we saw in the end was that we could train regression models that were quite accurate. So this is a protein-specific regression model for PAB1. It's just one of the proteins from the scan. You have in the plot on the bottom the predicted functional score on the y-axis. I'm sorry, on the x-axis, and the observed functional score on the y-axis. On the y-axis, and what you can see is that the correlation is quite high. So the R squared value here is 0.86, and that suggests that our model is doing, and the R squared values that I'm showing here and in other places are going to come from cross-validation that we did in the model training. What you can see is that the model is quite accurate. So this told us that the features that we chose and the model training procedure that we're using is a sensible one, and it also has one other interesting corollary, which is that we can use the model to fill in missing data or complete a deep mutational scan. So this is the PAB1 data laid out in a heat map, sort of like you saw before, where along the x-axis are positions of the mutagenized region of PAB1, and on the y-axis are all the different amino acid substitutions you could make. And yellow means more deleterious, blue means less deleterious. So what you can see are a bunch of white boxes. Those white boxes are missing data. Missing data is a part of really any deep mutational scan. Usually there's between 5 and 50% missingness for a variety of reasons. But since we had such an accurate predictive model, what we figured we could do was actually fill in the missing data using our model. And of course this assumes that the data that we're missing is no different in kind anyway than the data that we have. And I think that's a reasonably good assumption. But in any case, we can use these protein-specific models to complete deep mutational scans, which is useful if our goal is to use deep mutational scan to operate on a particular protein and make predictions. So this is just a plot of R-squared values for a number of different protein-specific models that we made, and you can see that some of them are about as good as PAB1, others we don't do so well at, and we're not sure here whether this is a result of data quality or some deeper sort of biochemical truth about the protein that makes it more sensitive to mutations or less predictable. But in any case, this is a good sign that our modeling process is working well. So we next moved on to a more challenging problem where we wanted to train a global regression model. So here, our goal is to now mash all the training data sets together and train a model that can predict, and then we de-identify the training data, so the model can't learn from which protein the training data came, right? And now we want to train a general predictor of variant effect from these 11 deep mutational scanning data sets. And again, we're trying to train a regression model, meaning we want to predict the actual functional score of each mutation. And this, I think, is a really, really hard task, if you think about it. What we're sort of asking is, can we learn how to predict the magnitude of effect of a mutation accurately across a range of proteins that are pretty much unrelated? So it turns out that we can train a global regression model that performs pretty well in cross-validation. So what you're seeing here is the R-squared from cross-validation for the model. It's about 0.7, which is satisfyingly high. A question that we were interested in asking is, what are the important features of this model? In other words, what features are the most informative? And we find, just like many others have found, that solvent accessibility and conservation are the most important features in our model. So that's really no surprise. Now, when I show you an R-squared value from cross-validation, it's nice to see, but it's not really what we actually would want this model to do, which is make predictions on a data set that the model has never seen at all. And so to sort of see what would happen if we tried that, we trained the global model where we left one protein completely out of the training, and then asked the model, the model that was trained on the other 10 proteins, to predict what would the functional effects of that one left out protein. And we see that we can do it, sort of. So we get an R-squared value of 0.4 for beta-lactamase. Other proteins are sort of in the range. A few are better, many are worse than this one. And so what this means is we can predict with some accuracy the functional scores of a protein. And I guess this model would work on sort of any protein that you fed it. But I don't think we can claim that this is really working well enough to say we've solved the magnitude effect prediction problem and can just go out and predict the effective magnitude of any substitution in any protein. But I do think it hints at how, as we refine our modeling procedure and get more data, we'll be able to, maybe we will actually be able to tackle this problem, which would be really fantastic and provocative if we could do it. So given that, we retreated to a slightly more modest task, which is now just training a classification model. So again, a global model works on all the protein data sets that we collected, intended to predict the effects of mutations on any protein. But now we're just going to classify proteins into either variants into either deleterious or neutral. OK. The first thing we had to do in order to train such a model is discretize our deep mutational scanning functional scores. And we did this using the synonymous distribution of variants available in these deep mutational scans. So we've been talking about the non-synonymous variants that we get from deep mutational scanning, but we also, in many cases, get synonymous variants sort of for free based on how the libraries were created. And you can see the synonymous score distribution on the right-hand side here for the ubiquitin deep mutational scan. Just as we would expect, it's a very narrow distribution because synonymous variants don't tend to have functional consequences. So we used a bootstrap to construct a 95% confidence interval on the synonymous variant distribution, and then used that confidence interval to classify the non-synonymous variants as either damaging or other. So now we have our training data for a classification model. So the classification, just to show you what the functional score discretization looks like for a number of the other proteins in the training data set, you can see that in general we have more damaging than other mutations to different degrees with different proteins. So this can be a reflection of data quality, and there are some sort of deep issues that we don't have time to get into here. But this is what we saw. So we went ahead and trained a global classification model and saw that it was 88% accurate in cross-validation. Again, solvent accessibility and conservation are the most important features. But moving on to the sort of gold standard test, which is how well does the model work on data that the model hasn't seen at all, we left out each of the 5 proteins that you see there, trained classification models, and this is just the accuracy in cross-validation for those models. And what you can see is that no one data set makes a big difference, or no one data set when left out induces a big drop in model accuracy, which is a good thing. When we then plot the model accuracy on prediction on the unseen data set, we see that we get quite high accuracy in most cases, which is really great, right? That tells us that our model is actually successful in predicting the effects of variants, or classifying variants into either damaging or neutral for a protein that the model has never ever seen, which is great. So we called our best model Envision, and I'm just going to show you a few more comparisons now between Envision and other predictors on a number of prediction tasks. So this is a comparison. Ooh, that slide got a little messed up. Sorry. What the y-axis is supposed to say is AUC, so it's area under an ROC curve, which is another metric of model accuracy. And what you can see here is that for the WW domain dataset, Envision performs way, way better than three other commonly used models, which is a very nice thing to see. We wanted to use an independent dataset that we didn't collect to validate our model, and we turned to that dataset to the Protein Mutant Database, which is a database of over 100,000 mutations curated from the literature by a group in Japan. And these mutations have been studied in a biochemical way, so we know what their biochemical effects are, and they were classified into a bunch of different classes, ranging from neutral to complete loss of function. So this is a nice dataset to test the performance of Envision on. For this test, we took the neutral mutations as neutral, and then anything classed as deleterious, strongly deleterious, or complete loss of function as damaging. And then we compared Envision's prediction capability to the prediction capability of a number of other predictors, and this is just an ROC curve showing what happens. So Envision is more accurate than every other model that we tested, except for one, and that's SNAP. SNAP is slightly more accurate than Envision, but it's also not a fair comparison because SNAP was trained on this particular PMD data set. So it's sort of hard to make a comparison in that case. In any case, what this tells us is that from 9 protein deep mutational scans, we can train a model that is at least as good and in many cases better than other variant effect predictors out there. So that made us quite happy. So the final thing we wanted to know was whether Envision could improve pathogenicity predictions. So we've been talking about biochemical effects up till now, right? That's sort of what Envision is for. That's what the data sets it's trained on are biochemical effect data sets. Most other models are trained on predicting variant pathogenicity from databases like ClinVar that have a bunch of mutations that are classified as either pathogenic or not. So we examined Envision's predictions on pathogenicity or on pathogenic variants in ClinVar, and this is an ROC curve showing you that Envision doesn't do very well in pathogenic variants, which is no surprise because that's not how we trained it. But what you do see that's interesting is that if you add Envision to the best performing model in our hands, which is CAD, you now get a combined model that's even better than CAD and certainly better than any of the other models. And this tells us that adding sort of biochemical effect predictions to these pathogenicity predictors can actually gain us a little bit of extra mileage in predicting variant effects, which is a cool result, I think. Okay, so that's Envision, our crack at variant effect prediction, which I think has turned out quite nicely. We're hoping to make it publicly available in the next month or so via a website and a paper. So finally, I want to use the last 15 minutes or so, 10 minutes or so of my talk to talk about this issue of combining variants at multiple loci. So up until now, we've been talking about individual proteins, right? But we know that interactions between variants at different loci are important for human disease. So if we think about genome-wide association studies, there's sort of this issue of missing heritability has come up. And what we mean here is that when we look at the SNPs that are considered significant in most GWAS, they don't, if you add them up, explain all the variation that, or all the heritability, sorry, of that particular disease or trait. And the missing heritability, there are many hypotheses about where it is, but one idea is that it could be combinations of variants at multiple loci that cause this missing heritability. In GWAS analysis. So another example is in cancer, where in cancer, many mutations are acquired by cells, but then go on to become cancerous, and the order in which these mutations are acquired and sort of how they are permuted and combined can have big implications for how aggressive the cancer is and how you can treat it. So we would very much like to be able to study interactions between loci in an experimental way, but this turns out to be hard to do. So if we think about a sort of simple experiment we might want to do, where there were 5 loci and 10 alleles at each locus, we would have 1,000 combinations to make. And this is too many to make using sort of certainly using traditional methods. But even if we turn to sort of the gold standard way of editing genomes now, which is Cas9, and in case you've been under a rock, Cas9 is an RNA-guided endonuclease that can make site-specific double-stranded breaks, and then we can use homology-directed repair by introducing a template with homology arms to repair those breaks and put in variants that we're interested in. So this is great. All this is great. The problem is that Cas9 is not particularly efficient, especially when it comes to HDR. And if you start thinking about editing multiple loci, this inefficiency becomes prohibitive very quickly. So imagine that we had 2 really highly efficient Cas9 guides and HDR cassettes where we could get 10% HDR at each locus, right? That means that only 1% of our cells would be doubly edited, that is, have edits at both of the 2 loci we were interested in. And really 1% is not enough 1% is not a pure enough population to do the types of biology or screens that we would like to do to study how variants at each locus influence the effective variants at the other. So what we thought about was trying to make a reporter to detect successful genome editing that would be scarless on the message and or protein level, because we don't want to give up our advantage of endogenous locus editing completely, that would be site-specific and multiplexable. So in thinking about this, we came up with a design of a synthetic intron that could mark editing at any locus that was near a coding region. And the idea of the synthetic intron is that we would have a repair template that would contain our desired edit or edits, could be elaborated edits, and then we would have a synthetic intron that would sort of either replace a natural intron or just be introduced into an exon de novo, and that synthetic intron would be able to express a fluorescent marker. So that then what would happen, and I'll talk about the design of the intron in a minute, so don't worry about it too much, but if we can make such an intron work, then what would happen is that our successfully edited cells would become fluorescent, and we could then flow sort for the edited cells. If we think about how this would work in a multi-locus fashion, it's pretty easy to see that we could mark each locus with a different color so that multiply edited cells would be flow sortable based on their multiple coloration or multiple fluorescence. So that's the basic idea behind what we're trying to do. And the design of the synthetic intron that we came up with overcomes a few of the key problems that come up when you start thinking about what you would need to make an intron that could express a protein. And so this is the design of the intron that we made. We started with actin, and we started with the 3rd intron of actin, and co-opted the 3rd intron splice donor and splice acceptor sites from the actin intron 3. Now, introns are not particularly stable. So in order to stabilize our synthetic intron, because they get spliced and then debranched and rapidly degraded. So to stabilize our synthetic intron, we stole a couple of viral sequences, RNA sequences that generate these big knotted and helical structures that prevent degradation by exonucleases. So this was our way to stabilize the intron, the synthetic intron. So now that it's stable, we've got to get it out of the nucleus so it can encounter a ribosome and be translated. So to do that, we stole another viral sequence, which is a constitutive transport element that gets RNA sequences out of the nucleus. Finally, we added an internal ribosome entry site and an EGFP coding region, which, if all these other elements work right and the intron is stable and gets out of the nucleus, should enable the ribosome to find and translate this sort of synthetic message that we've created. Now, we interrupted the actin ORF with the synthetic intron, and also appended an mCherry as sort of a reporter for correct actin splicing. So the first thing we tried was seeing if this synthetic intron would function on a plasmid. So I'm showing you here untransfected cells. This is flow data where you have the mCherry signal on the y-axis, the GFP signal on the x-axis, and then histograms of each signal so you can kind of see where the points are in the scatter plot. We see that there's not much signal from untransfected cells. That's good. When we transfect cells with actin and its natural third intron, just to make sure nothing gets screwed up there, we see the expected mCherry signal, but no GFP signal, so no synthetic intron. When we now transfect actin with the synthetic intron in it, we very pleasingly see that we get both mCherry signal and GFP signal. And this suggests to us that our synthetic intron is getting transcribed and translated. But it could be that the message is never being spliced, right? And that the full-length message with the synthetic intron in it sort of gets out of the nucleus, and then that iris signal just leads to GFP expression, even though there's no splicing, which would be a very bad thing if it was happening. So what we did was delete just the splicing elements from our synthetic intron construct, and that's now shown in the center panel. And what you can see is that when you delete just the splicing elements, you now get, not only do you get no mCherry expression, because it's now out of frame, but you also get no GFP expression, indicating that, or at least suggesting, that splicing has to happen in order for us to see the GFP signal that marks, or that indicates that the GFP signal that comes from the synthetic intron. So, so that sort of told us that things were working as expected on the plasmid. And so the next step then was to ask, could we actually see this, make this work in a genomic context? So we did the obvious thing, which is take our actin construct and use Cas9-mediated HDR to recombine this construct into the genome. So now what we have is a population of cells in which we replace the third intron of actin with our synthetic intron cassette. Note that mCherry has been completely removed from this genomic, genomic synthetic intron construct. So we isolated a bunch of cells, grew them up for 4 weeks to dilute any remaining plasmid, which of course will still express both markers, and then isolated single cells to create clonal lines. This is how the parental cells look when we flow sort them. So just as you would expect, there's no mCherry signal and there's no GFP signal, which is comforting. Now, if we look at the so when we look at the clones that we get two phenotypes. One phenotype is lots of GFP signal, no mCherry signal. This we interpret to mean that these cells likely have our synthetic intron recombined into the correct location in actin, where the synthetic intron is now being transcribed as part of the initial transcript, spliced out, and then exported from the nucleus, and the GFP translated. So we get some clones that look like this. We also get some clones that look like this, where we see both GFP and mCherry. And we were expecting these types of clones. And this is just essentially random integration of the plasmid into the genome, right? Where the plasmid, of course, contains the synthetic intron and also the actin mCherry fusion. So we were expecting to see this. And comfortingly, what's true is that of the 10 clones that we've isolated so far, 8 are GFP positive, mCherry negative. That is the thing we want. And 2 are GFP positive, mCherry positive. That is the thing we don't want. But in any case, I think this demonstrates the basic idea works, right? We can make a synthetic intron that's stable, that can express a gene, which is, I think, a pretty cool thing. And we can use that gene, that marker, to select cells that have been edited at the locus in question. So now what we're doing, is basically showing that we can use this at multiple loci, and that we can actually put in libraries at each locus and get out the desired product, which is a set of cells that have been edited at multiple loci simultaneously. So so that's the synthetic intron project. And really, that's what I wanted to say today. So I hope I've convinced you that there are a number of sort of interesting methods that can be used to try to make connections between the sequence of a protein and its function. I also showed you a little bit of data about how that sequence function maps can be useful in interpreting variation we find in human genomes. Deep mutational scanning has kind of found its legs and I think is becoming widely applied, and we're excited to sort of see where it goes. I told you about Envision, which is an accurate variant effect predictor that we've developed, which is trained on a different kind of training data than other variant effect predictors, and we argue could be useful for different purposes. And finally, I told you about this synthetic intron business, where our goal is to construct multi-locus libraries of variants. And so with that, I want to close by thanking the many people who did this work. So in my lab, the deep mutational scanning work was sort of spearheaded initially by Jason Stefani. The BRCA1 data that I showed you was generated primarily by Leah Starita in Stan Field's lab, and that project was also a collaboration with Jay Shanduri. For Envision, the computational predictor, that project was spearheaded by Vanessa Gray, a grad student in my lab, in collaboration with Ron Haas and with assistance from Jens Liebeck in Jay Shanduri's lab. And then the synthetic intron story actually started as a rotation project from a very brilliant rotation student, Molly Gasparini, in the Genome Sciences graduate program, and it's been continued by a postdoc in my lab, Kenny Matrejak. So with that, I'd like to thank all of you for your time and attention, and say that I'll be very happy to answer any questions that you might have.
Great, thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Right now, it looks like we don't have any submitted questions. So with that, we can go ahead and wrap up this presentation. Today's webcast will be available for on-demand viewing through November 2016. You will receive an email from LabRoots alerting you when the webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.